Before I start today's topic, let me first explain or clarify your doubts that have been asked by you. The first doubt is, what is the difference between bilobed and dithecus? So let me explain what is bilobed and dithecus. So I have explained the anther of angiosperm is bilobed. If you recollect this diagram, then you can see that here there are two projections. The whole structure is the anther, and these are two projections, or rather I can show you like this. So these are the two projections, okay, two flat projections. Now when we take the transverse section, we see that in each lobe, this is one lobe, this is another lobe. So in each lobe, so okay, so here I had shown you two chambers, okay, so two chambers. So each lobe, this is one lobe, this is one lobe, this is another lobe, and in each lobe two chambers are there. You can see this is one chamber, this is another chamber. So the chambers are known as theca, okay. So it is dithecus. Each anther consists of two lobes, two projections, and each projection consists of two chambers, okay, and the chambers are known as theca. So that's why it is dithecus, bilobed, okay. And in each theca the sporangium is present, microsporangium is present. In each theca, a microsporangium is present. So total how many theca are there? 4 theca are there. And so in total how many microsporangium are there? 4 microsporangiums are there. I think you got it. Now come to the next question. The second question, what is the role of the connective? Okay, let me show you again the diagram. Okay, so here you can see, here I have drawn a diagram which consists of parenchymatous tissue, okay, and this tissue helps in connects the two lobes. It helps in connecting the two lobes. Besides that, this is the reason where the filament is attached to the anther. Filament is attached to the anther. So to this what are present inside the anther? The anther, inside the anther, the sporangium are there, and in the sporangium the sporogenous tissue are there, and they need supply of water, supply of nutrient for the division of cell, for the growth of the cell. Then where from they get this nutrient? They get the nutrient through the connective tissue, and here through this connective, the vascular tissue are supplied to the anther. You know, vascular tissue means xylem and phloem. So xylem carries water and mineral, and phloem carries the food. So they bring the food and the water and mineral for the survival of, for the growth of the cells inside the anther through the connective. So I think it is clear to you. Now let us start today's topic. So today we will discuss, yesterday we discussed the structure of anther, and the anther consists of two lobes. Each lobe has two theca. Each theca has one microsporangium. Okay, and in the microsporangium, okay, so this is one microsporangium. Okay, now in the microsporangium, the sporogenous tissue are there. The microsporangium contains sporogenous tissue. Okay, so they are filled with sporogenous tissue, and I have already discussed the membranes that surrounds the microsporangia. Sporangia is the plural of microsporangium. Okay, now this microsporangium that contains the sporogenous tissue, the cells of the sporogenous tissue undergoes microsporogenesis. Okay, so now let us discuss the process microsporogenesis. Go on writing, children. So what is microsporogenesis? It is the formation of microspore tetrad from a microspore mother cell. Okay. Now what is microspore mother cell? So I am giving you a schematic diagram. So you can write the if this is asked in the exam, you can write in schematic diagram. That will be the more appropriate way, convenient way of presentation. So the microsporangium contains sporogenous tissue. Okay, and this sporogenous tissue each each cell of sporogenous tissue develops into microspore मदर सेल. ओके, तो ये जो आपका माइक्रोस्पोरेंजियम, दिस इज माइक्रोस्पोरेंजियम विच is composed of the cells, and so these are the sporogenous tissue. Now each cell can give rise to a microspore mother cell. So now this is the microspore mother cell. This is the single cell of sporogenous tissue, and you know that since the whole plant body is diploid and so far no meiosis has been taken place. In class 11th you have studied that there are two types of cell division, mitosis and meiosis. थोड़ा सा मैं साइड में ये बता रही हूँ, आप साइड में कहीं पर लिख लो. Don't write on the main portion because I have to continue this. So somewhere write down mitosis. In mitosis, if the parent cell is 2n, this is parent cell, then it divides into two daughter cells which are also having 2n number of cell. So if the parent cell is diploid, then daughter cells are also diploid, or if the parent cell is haploid, then the daughter cells will also be haploid. That means the number of chromosome remains same in the daughter cell. So now come to the meiosis. So, have you written this? Now in case of meiosis always takes place in diploid cell. Okay, and these cells are known as, these diploid cells are known as meiocytes. Meiocytes. Any cell that can undergo meiosis is known as meiocytes. So what are the meiocytes? Meiocytes are the gamete mother cell. Because meiosis takes part in the meiosis takes place in the diploid germ cells to form the haploid gametes. Okay, diploid germ cell, yani germ cell means here we can take the sporogenous tissue, and in case of human being का example मैं दे रही हूँ आपको, that is the cell of the testes and the cell of the ovary. Okay, that we will discuss in the next chapter. But ये जो सेल होते हैं जो कि गैमेट में डेवलप होते हैं उसी को कहा जाता है मियोसाइट्स. सो व्हाट आर दे इन जनरल? दे आर द गैमेट मदर cell. Okay, the cell in which meiosis takes place is called meiocytes. They are always diploid cells. The meiocytes are also known as gamete mother cell because these are the cells which divides to form the haploid gametes. So in meiosis what happens? the parent cell divides twice, that is meiosis 1, in which two daughter cells are produced which are haploid. Then it undergoes meiosis 2 and it forms 4 haploid daughter cells. So these are the daughter cells. So when it takes place in the gamete mother cell, then these are the gametes. Okay, I think it is clear to you. So इसको थोड़ा साइड में हम रख देंगे. Now come to the main topic. Have you all written this in your copy? Now come to the our main topic, that is microsporogenesis. So this is microspore mother cell. So its ploidy is 2n. Okay, remember, wherever you are doing any process, any division, in each and every step, don't forget to write the ploidy. Okay, now this microspore mother cell undergoes meiosis, and as you know that meiosis takes place in 2 steps, that is meiosis 1 and meiosis 2. So in meiosis 1, after meiosis 1, it forms microspore dyad, means it is divided to form the 2 daughter cells, but they are attached to one another, they are in contact with one another, so they are known as microspore dyad. Okay, so this microspore mother cell is otherwise known as pollen mother cell. This microspore mother cell is otherwise known as pollen mother cell or PMC. PMC. Okay, so this PMC undergoes fast meiosis and it forms 2 cells which are each of them are N. They are N, means haploid. Now this microspore dyad undergoes meiosis 2 and it forms 4 cells. Each of them divide to form 2 more cells. Each of them are haploid. So all of them are haploid or N. Okay, so this is called microspore tetrad. And this process of formation of microspore tetrad from the microspore mother cell is known as microsporogenesis. Now when the anther matures, when the anther matures, after maturity of the anther, it undergoes dehydration, matures and undergoes dehydration Then what happens? These microspore tetrad, the microspore tetrad detach from each other. They detach from each other. ये एक दूसरे से separate हो जाते हैं. They dissociate from each other and develop into pollen grains. So each of them develop into pollen grains. So how many pollen grains are formed? 4 pollen grains are formed, and these 4 pollen grains are together known as pollen tetrad. Pollen tetrad. So each of them is pollen grain, and the ploidy of pollen grain is N. Remember, the ploidy of pollen grain is N. Okay. So when the anther matures, dehydrates, then the microspore dissociate from each other and they develop into pollen grains. So in a microsporangium, how many pollen grains are produced? The pollen grains are similar to the, is it similar to the number of sporogenous cells present in it, or something else? Remember here, each cell is a microspore mother cell, and each microspore mother cell gives rise to, or you can say it divides to form 4 microspore and further which develops into 4 pollen grains. तो यहाँ पर आप याद रखो बच्चों, इसमें न्यूमेरिकल्स पूछे जाते हैं. एक one cell of sporogenous tissue forms 1 microspore mother cell, and 1 microspore mother cell forms 4 microspore or 4 pollen grains. So at the end I am going to give you the homework. I think it is clear to you. Now when the anther dehisces, when the anther dehisces, the pollen grains are released. But I have explained you that each microspore develops into a pollen grain. That means during the course of this development, some changes must be taking place in the microspore. So let us see what changes makes the microspore, or what changes converts the microspore into a pollen grain. So now development of microspore into pollen grain. Very important, children. So each and every word is important here. Okay, whatever I am writing here, saying here is very important. So now let us take one microspore. Each microspore has 2 covering, outer covering, and a single nucleus. So this is a microspore. Okay. Now this microspore, it grows in size. Okay, and it stores the food materials and water in the vacuoles. So now this microspore develops into a structure, or it grows in size and develops the vacuoles which stores the nutrient. Okay, and this is the nucleus. Okay, now this nucleus undergoes unequal division. It undergoes, the nucleus undergoes unequal mitotic division. तो आपका जो न्यूक्लियस है अभी वो माइटोसिस होगा, तो इसका प्लॉइडी क्या था, इट्स प्लॉइडी is n. So this is also n. So now this n cannot divide to form n by 2 n by 2. It cannot divide further by meiosis. So now whatever division will take place, that will be mitosis. Remember children, once the cell is haploid, means it cannot undergo meiosis, it can undergo mitosis only. सिर्फ mitosis होगा. So now this nucleus undergoes mitosis, and that is unequal. So here asymmetric spindles are formed. You know that during cell division, the spindle fibers are formed to hold the chromosomes. So here are the chromosomes which are held by the spindle fiber, and these spindle fibers are known as asymmetric spindle. Okay, सो ये जो स्पिंडल होते हैं छोटे बड़े होते हैं, इसीलिए उसको एसिमेट्रिक स्पिंडल कहा जाता है. दे आर नॉट सिमेट्रिकल. नाउ दिस न्यूक्लियस divides completely to form two cells, two nuclei, and as I have said you that it divides into two nuclei, but the division is unequal. So one of the nucleus is larger in size and other one is smaller in size. The larger sized nucleus remains inside the complete cell, means this cell, the cell inside which the division has taken place. So this is vegetative cell. Now inside the vegetative cell only, the small nucleus is floating, and that gets surrounded by the cytoplasm. ये जो न्यूक्लियस है, एक बड़ा एक छोटा, ठीक है, बड़ा वाला जो ये बड़ा सेल का साइटोप्लाज्म में रहेगा, और जो छोटा वाला है दैट गेट्स सेपरेटेड बाय द सेल वॉल, and it forms another small cell which is floating in the cytoplasm of vegetative cell. ये जो बड़ा सेल है उसको हम बोलते हैं वेजिटेटिव सेल, और ये जो छोटा सेल है उसको हम बोलेंगे जेनरेटिव सेल. Okay, and it has two outer layers. I have not drawn here. So now this is the pollen grain. Okay. So this pollen grain has two cells. Till now we have seen. But in future what happens? This pollen grain, it may be released from the anther in two-celled states, very important children, or three-celled states. ये जो हमने बताया ये कौन सा स्टेज है? इट इज़ टू सेल स्टेज, दो सेल है, वेजिटेटिव सेल एंड जेनरेटिव सेल, दो ही सेल है. सो ये दो सेल क्या क्या है? द वेजिटेटिव सेल and generative cell. But in three cell stage what happens? In three cell stage what are the cells? The cells are vegetative cell and so here they are one in number, this is also one in number, but in three cell stage one vegetative cell and male gametes two in number. So what happens in two cell stage? If this is the pollen grain, then this is vegetative cell, this is generative cell. But in three cell stage, the vegetative cell remains as it is. So this is vegetative cell. Then the generative cell divides into two male gametes. So the pollen grain may release from the anther either in the two-celled stage or in three-celled stage. So often in exam it is asked what are the two cells and what are the three cells. Okay. Now the generative cell is spindle shaped and remember children, in 60% cases the pollen grains are released in two-celled stage. Okay, and in only 40% cases the pollen grains are released in three-celled stage. Okay, I think it is clear to you. Now let us discuss the structure of a pollen grain. Structure of pollen grain. So each pollen grain is having this structure. All of you draw this diagram. Draw a big diagram. Okay, so it has two layers. The outer layer is exine and the inner layer is intine. Okay, and these are the reason you can mark here, you can observe here. These are the reason where the exine is discontinued. So exine is not forming a continuous layer. So at some places, maximum 3 places only, maximum 3 places, the exine is discontinued. So, and this forms the germ pore. So germ pore, it is the region where only intine is present. Okay, so the outer layer is exine, the inner layer is intine, and inside which two cells, as in 60% of cases two cells are there. So I will discuss two cells. Two cells are there. One is vegetative cell and other is generative cell. Okay, so if in the exam you are asked to draw the structure of pollen grain, then draw this diagram, label it, and also write the composition and function also. So let me tell you what are the composition and function of each and every part. So each pollen grain is spherical, and it is about and 20 to 50 mm diameter. It is, that size is not so much that much important. Now come to the various parts. It consists of exine. Exine is the outermost covering made up of sporopollenin. Remember, it is made up of sporopollenin. Okay, and this sporopollenin is highly resistant to, this is highly resistant to the strong acid and strong acid and alkali can withstand or can resist high temperature, and no enzyme can degrade the sporopollenin. So no enzyme can digest the sporo. तो इसको लेकर आपके मन में एक सवाल उठना चाहिए. मैं आगे जा रही हूँ, मैं देखती हूँ आप मुझे पूछ रहे हो कि नहीं. ओके, पर्टिकुलरली इसी पॉइंट को लेकर आपके दिमाग में एक क्वेश्चन आना चाहिए. नाउ नेक्स्ट इज योर intine. Its function, let me tell its function. It provides protection. It can be the pollen grain. The pollen grain can be preserved as fossil due to presence of sporopollenin. Okay, so this is about the exine. Now come to the intine. So intine is the inner layer. It is the inner covering, rather I can say. It is thin and it is made up of cellulose, and it is thin and made up of cellulose and pectin. Cellulose and pectin, and it is a continuous layer. It is a continuous layer. It is a but this one is not continuous layer. Exine is not a continuous layer. Okay, why? Because at the region of germ pore it is discontinued. Okay, now what is the function of intine? It also provides protection to pollen grain and helps in the formation of pollen tube. Okay, so I will tell you what is pollen tube. जब पोलन ग्रेन आपका जर्मिनेशन होता है, ये है पोलन ग्रेन, जब ये जर्मिनेशन होता है तो इसमें क्या होता है आपका, जहाँ पर आपका जर्म पोर होता है, यहीं से आपका इंटाइन it emerges out along with the nuclei that we will discuss later on, and this is called pollen tube. Okay, now the next part is germ pore. It is the portion on the covering where sporopollenin, it is single sentence, pollenin is absent. So what is the function of germ pore? The pollen tube emerges out through it during pollen germination. Okay, so this is the function of pollen tube. Now come to the vegetative cell. Bigger in size, dense cytoplasm is present, and its function is to provide nourishment to the generative cell. जेनेटिव सेल छोटा सेल होता है जो कि आपके वेजिटेटिव सेल के अंदर फ्लोट कर रहा होता है. तो वेजिटेटिव सेल में जो साइटोप्लाज्म है, दैट साइटोप्लाज्म प्रोवाइड्स नरिशमेंट टू द जेनरेटिव सेल. नाउ कम टू जेनरेटिव सेल. Now the generative cell, it divides mitotically to form two male gametes. Okay, so now where what was our aim? Our aim was, इतना सारा जो हम पढ़ रहे हैं, मेल रिप्रोडक्टिव स्ट्रक्चर पढ़े, फिर उसके बाद माइक्रोस्पोरोजेनेसिस, ये सारे जो पढ़े, हमारा एम क्या था? टू गेट द मेल गैमेट. सो अल्टीमेटली मेल गैमेट कहाँ से मिलता है? बाय द डिवीजन ऑफ द जेनरेटिव सेल, एंड दैट टू बाय माइटोटिक डिवीजन. ओके, नाउ हियर वेरी वेरी इंपॉर्टेंट, मैं बॉक्स बना के लिख रही हूँ. द पोलेन ग्रेन is known as male gametophyte. Okay, very very important. पोलेन ग्रेन में मेल गैमेट फॉर्मेशन हो रहा है and it carries the male gametes, so it is known as male gametophyte. Now the pollen grains of many species causes allergy. Some of you may also be having the allergy to the pollen grains. So the allergies causes allergies and bronchial affliction. Okay, bronchial inflammation, secretion of mucus from the bronchus, the bronchus swells off and we feel difficulty in breathing. Okay, and in some cases it leads to severe or chronic respiratory disorders. Respiratory disorders like asthma, bronchitis, etc. Okay, and in your textbook one example is given, that is Parthenium. So example is Parthenium, the scientific name, and the common name is carrot grass. The common name is carrot grass. So the pollen grain of this one causes allergy, and it was came to India. It came to India as contaminant with imported wheat. जो हम बाहर कंट्री से व्हीट ला रहे थे, तो व्हीट के साथ ये हमारे यहाँ आ गया था. एंड बिकॉज इट हैज़ नो नेचुरल प्रेडाटर्स, इसका कोई कंपिटिटर नहीं है, कुछ नहीं है, तो ये आसानी से ग्रो कर जाता है. एंड इट्स पोलन ग्रेन कॉजेस एलर्जी इन मैनी पीपल. नाउ व्हाट आर द यूजेस ऑफ दिस पोलन ग्रेन? फॉर द ह्यूमन बींग के लिए किस काम में ये आता है? सो इट्स इकोनॉमिक इम्पोर्टेंस ऑफ पोलन ग्रेन. सो ये कैसे ह्यूमन बींग के लिए, अब इसमें तो मेल गैमेट होता है, वो रिप्रोडक्शन में हेल्प कर रहा है, दैट इज द मोस्ट इम्पॉर्टेंट थिंग. बट बिसाइड्स दैट, इन व्हाट वे इट इज हेल्पफुल फॉर द मैनकाइंड? द पोलन ग्रेन्स are rich in nutrients, okay, and that's why hence the pollen tablets are taken as food supplement. Okay, this is one use. Next, in western countries, the syrups are also available, and the pollen consumption increases the performance of athletes and race horses. ओके, सो ये दो काम में हमारे आते हैं. बिसाइड्स हेल्पिंग इन क्रॉप डेवलपमेंट, क्रॉप मैनेजमेंट, it is helpful in this way. Now another most important thing I am writing here, the landing platform for for pollen grain is स्टिग्मा. ओके, जब पोलन ग्रेन एक फ्लावर का अंतर से निकल के जाता है तो दूसरे फ्लावर का या फिर सेम फ्लावर का स्टिग्मा के ऊपर लैंड करता है. ठीक है, इट फॉल्स ऑन द स्टिग्मा, सो इट इज द लैंडिंग प्लेटफॉर्म फॉर द पोलन ग्रेन. So this pollen grain has viability, means how long they are having the ability to fertilize. The duration the ability of the pollen grain to fertilize is called viability. Okay, so this viability is different for different flowers. Okay, and this viability is highly variable depending upon the temperature and humidity. So now in some cereals and cereals like in some cereals like rice and wheat, it is maximum 30 minutes. Wheat, the pollen grain loses viability within 30 minutes. That's why the rice and wheat are grown in mass. ओके एक छोटा सा स्पेस में बहुत सारे प्लांट को नज़दीक, मतलब वेरी क्लोज टू ईच अदर दे आर ग्रोन. ओके, सो दिस इज़ वेरी वेरी इंपॉर्टेंट. ये बच्चों, ये जो पॉइंट मैं बोल रही हूँ ये सारे इम्पॉर्टेंट है. नाउ नंबर 2, जो एग्जाम्पल आपके बुक में है उतना आपको याद रखना है. नाउ सब मेंबर्स ऑफ प्लांट फैमिली हम पढ़े थे क्लास 11th में. तो हम सोलानेसी फैमिली पढ़े थे. सो बिसाइड्स सोलानेसी फैमिली 2 अदर फैमिलीज आर देयर. मेंबर्स ऑफ फैमिली, not all members, some members of family Leguminosae, okay, then Solanaceae and Rosaceae. Okay, so they have the viability for several months. ये months में होते हैं इसका viability. Okay, and this pollen grain can also be stored for future breeding program. So storage of pollen grain. So it is stored in very low temperature of liquid nitrogen. Okay, it is preserved by cryopreservation. Means very लो टेम्परेचर में स्टोरेज किया जाता है एंड इन लिक्विड नाइट्रोजन which is having temperature -196 degree Celsius. Okay, so these pollen grains are stored in a place called pollen bank. This is called pollen bank. So why it is required what is the advantage of storing the pollen grains? To facilitate the crop breeding program. ओके, क्रॉप ब्रीडिंग प्रोग्राम में मान लो हमको एक डिज़ायरेबल कैरेक्टर चाहिए, आर्टिफिशियल हाइब्रिडाइजेशन करना है, तो उस केस में हमको पोलेन ग्रेन, स्पेसिफिक पोलेन ग्रेन चाहिए. तो फॉर दैट पर्पस द पोलेन ग्रेन्स आर स्टोर्ड एट वेरी लो टेम्परेचर ऑफ -96 degree Celsius, which is maintained by the liquid nitrogen, and such type of preservation at very low temperature is known as cryopreservation, and the place is called pollen bank. Like seed bank आपने सुना होगा, उसी तरह ये pollen bank है. ठीक है, तो आज के लिए बस इतना ही. Now take the homework. सोच के करना. ठीक है. if an anther has 100 microspore mother cell in each sporangium, then how many pollen grains can be produced from such anther, and also write the number of male gametes will be produced from this anther. So this is the only one question, but आपका माइंड का एक्सरसाइज बहुत करना पड़ेगा विथ कैलकुलेशन के साथ आपको इसका आंसर देना पड़ेगा. सो फॉर दैट you have to go through the lesson, I mean whatever taught today, you have to go through, you have to listen me, okay, and then you answer it. Don't search in the Google and chat GPT time and again. I am telling you, okay. Thank you very much.